Hello, my dear ladies and gentlemen. Today I am going to discuss with you a brief history of genetics. So we can start from the inheritance of physical trait has been a subject of curiosity and interest for thousands of years. The ancient Hebrew and Greek as well as later medieval scholars describe many genetics phenomena and propose theories to account for them. Many of these theories were incorrect. Gregor Mendel, an Austrian monk who is usually considered the father of genetics, advanced the field significantly by performing a series of cleverly designed experiments on living organisms, garden peas. He then used this experimental information to formulate a series of fundamental principles of heredity. My dear ladies and gentlemen, Mendel published the result of his experiment in 1865 in a relatively obscure journal. It is one of the ironies of biological science that his discoveries, which still form the foundation of genetics, received little recognition for 35 years. At about the same time, Charles Darwin formulated the theories of evolution, and Darwin's cousin, Francis Galton, performed an extensive series of family studies, concentrating especially on twins. In an effort to understand the influence of heredity and various human traits, neither scientist was aware of Mendel's work. Genetics as it knows today is largely the result of research performed during the 20th century. Mendel's principles were independently rediscovered in 1900 by three different scientists working in three different countries. This was also the year in which Landsteiner discovered the ABO blood group system in 1902. Archibald Gerard described alkaptonuria as the first inborn error of metabolism. In 1909, Johnson coined the term gene to denote the basic unit of heredity. The next several decades was a period of considerable experiment and theoretical work. Several organisms, including Drosophila, melanogaster, fruit flies, and Neurospora cross-breed mold, served as a useful experiment system in which they study the action and interaction of genes. For example, H.G. Muller demonstrated the genetic consequences of ionizing radiation in the fruit fly. During this period, much of the theoretical basis of population genetics was developed by three central figures: Ronald Fisher, GBS, Haldane, and Swall Wright. In addition, the modes of inheritance of several important genetic diseases, including phenylketonuria, sickle cell anemia, sickle cell disease, Huntington disease, and cystic fibrosis, was established in 1944. Oswald Avery showed that their genes are composed of deoxyribonucleic acid. Probably the most significant achievement of the 1950 was the specification of the physical structure of DNA by James Watson and Francis Crick in 1953. Their seminal paper, which was only one page long, formed the basis for what is now known as a molecular genetics. Molecular genetics, we can define molecular genetics is the study of structure and function of gene at a molecular level. Another significant accomplishment in that decade was the correct specification of a number of human chromosomes. Since early in 1920, it has been thought that human has 48 chromosomes in each cell. Only in 1956 was the correct number